This is Cochranes of Oxford Orbit TM DNA RNA model kit reference 0079. Model shows a 12 base pair section of DNA in the process of transcribing RNA. So what we've got here is a model I've just bought, and it's got shows a DNA double helix. We've got the template strand, we've got the messenger RNA forming, and then we got the non-template strand. So I'm going to get these out of the packet. Just comes in a little plastic packet. It's got guanine, cytosine, thymine, adenine, uracil, different colors representing the different nitrogenous bases. And it's got, looks like, let's have a little look here. What have we got? We've got these black plastic pieces represent the sugar ring. So that's your ribose or deoxyribose sugar. We've then got different colors for the base that will be attached to the ribose molecule or deoxyribose molecule, and the different colors correspond to different bases. So blue is thymine, but you could have red, which would be guanine. So that's our sugars and bases. They've also got uracil if you're doing mRNA, and the other bases as well, cytosine and adenine. Right, let's have a little look. What else have we got? We've got a phosphate group. So we're going to need a phosphate because a nucleotide or a DNA or RNA nucleotide contains a phosphate, a sugar, the pentose sugar, and a base. But we've also got these little beauties that are hydrogen bonds. So they're what we're going to connect our bases together. All right, let's get this model constructed. Have a look what it looks like. Comes with a little stand, MDF kind of stand. Actually, it's not too bad that. It's cut nicely. And a little plastic support. So basically, this goes through the middle of the hydrogen bonds by the looks of things and holds the DNA molecule together. So let's get this put together. Okay, so this is the sugar phosphate backbone. It's 12 bases long. So we haven't attached the bases yet actually, but it has phosphate groups running down the outside. These yellow bands are the phosphodiester bonds, and then we have the ribose, deoxyribose, the pentose sugar coming off of each phosphate. Now attached to each pentose sugar, I've now got to connect the bases, so I'm just going to connect them in the order as it shows here in the instructions. A bit of my daughter's Play-Doh on there. All right, so red, blue, green, grey, blue, green, red, grey, grey, green, red, blue. So let's whack them in. Okay, now I've attached the bases, there's going to be another strand which I'm going to build over here. So now I have the complementary strand, and then we can go down. The green is cytosine, and that's going with guanine, the red. So I've got green and red, cytosine, guanine connecting, and I've got the grey, which is adenine, going with the thymine, the blue. Now, how do we bind them together? Well, how are two polynucleotide strands held together? Of course, if the bases are complementary, they form hydrogen bonds in between them. Now, we do have these representing the hydrogen bonds, so I'm going to go in and add those in. Okay, super. So I put the hydrogen bonding. Now, the only thing to say straight away about the model is it does have this little white bit here to represent the hydrogen bond. But as you know, some of the bases have 3 hydrogen bonds and some have 2, and you can't show that on this particular model. But for 20 quid, it's pretty good so far. Right, now let's attach it to the support stand. So here's the support stand, and I just need to slide this through the hydrogen bonds. And I guess this is going to allow it to hold up together when we start unzipping some of the DNA. Get in the hole. Okie dokie. All right. So there we go. It's not supporting very well there. So as we can see, it's bending at the moment. So obviously there's something else we got to do. 1, 2, 3, 4, 5, 6. They're all in. Slide the first 6 hydrogen bonds into the support rod. We've done that here. So these first 6 are into the support rod. Now what do our instructions tell us? Twist the DNA into a double helix by turning the top anti-clockwise. The DNA will shorten. Slide the 7th and 8th hydrogen bonds onto the rod. Ha! This is a very poor effort at getting the double helix on. And now apparently we'll be able to get on the 7th and the 8th. And the 8th. There we go. So with a little twisting, you then... we're turning it into a double helix. So you can see there the sugar phosphate bond. The double helix allows the DNA to rotate, and the sugar phosphate bond to protect the important codons, the triplet code that's held in here from chemical attack. So pretty clever stuff. And now on this one, I can get more on because it's in a double helix. We can get 8 of these hydrogen bonds on. Okay, so here is my DNA molecule. You can see it's got phosphate, and the black little plastic circles or rings are the sugars. So we've got our sugar phosphate backbone on either polynucleotide strand. And then here at the bottom on the support, it's twisted around in a double helix. And what's going to happen when transcription takes place is that an enzyme is going to attach at the start of a gene, and the strand is going to unwind, okay, just at that gene. So just at the part that we want to transcribe. Now, this is known as the template or antisense strand. Essentially, what's going to happen here is that free RNA nucleotides are going to come from the nucleoplasm or inside the nucleus and are going to attach by complementary base pairing to the template strand. So here, the white tube here in the key code in this manual is uracil. So uracil is going to bond by complementary base pairing to adenine, which is this grey tube. Why is it uracil? A common misconception, people think, well, uracil in RNA replaces thymine. That's correct. But then they get a little bit mixed up and think uracil binds with thymine on the DNA. It doesn't. Uracil binds with adenine, of course. Gets a little bit confusing. So the red tube is guanine. So this is my free RNA nucleotide with guanine. And that is going to join here, and it's going to bind there, guanine to cytosine. And then what I've got a cytosine binding to a guanine on the template strand, adenine, and adenine here is binding to thymine. There we go. Now, what happens next? The mRNA is nearly ready to go. It's nearly ready to leave via a nuclear pore and join to a ribosome. But first of all, the RNA polymerase is being moving down the mRNA molecule, or moving down the template strand, and it has been forming the sugar phosphate backbone by forming phosphodiester bonds that are going to hold the backbone together. So we're going to add those in now. It doesn't make that noise. That's purely optional. Enzymes don't normally make that noise. Okay, so there we go. Here is our 4 base long mRNA strand. Sugar phosphate backbone has been formed. Phosphodiester bonds have been formed. Condensation reactions, like most biological molecules, formed in condensation reactions. And the RNA polymerase plays a key role in that. So now this is ready to leave and exit the nucleus via a nuclear pore. Well, how do you like that model? This model is from Cochranes of Oxford. I believe they've closed down, but you can find these on quite a few websites. So I'm going to show you where you can get these on websites. Look, there's a really cool model here. Massive 3D model there. This cost me £20, but I have seen it going for £10 on some websites. So let's have a look where you can get yourself one of these.
Okay, so I've just Googled Cochranes of Oxford, the DNA thingy-me-bob that we've just been building here. Orbit model DNA RNA kit. And you can see it's going for £17.95, which is like £21 or £22 with VAT. I'm not sure how much postage they're charging, but it's a really cool model. I think it's worth it. But you can get it even cheaper. There's one over here for £10.66 from Rapid. And there you go. And that's including VAT. So that is dirt cheap. And for a pretty cool model to sit on your shelf. Good for teachers, good for or tutors for teaching transcription or just the structure of DNA. Anyway, hope you liked the video. Hit the like, subscribe. Let's go.
You're still here? It's over.